Good morning, everyone. I hope that we are fine. It's 8 hours. I think we can start. I understand that some among us have a class at 9 hours. I hope that we are fine. It's 8 hours. I hope that you can see my screen. So this lecture is a makeup for those of us who were not able to attend the other session. And it will be about DNA replication, and in particular, we're focusing on prokaryotes. Why am I emphasizing this? Because if you go on the internet or you're reading a book, you'll find that some of the information will be on prokaryotes, some of it will be on eukaryotes. So I don't want us to get confused. Our focus in this course is DNA replication in prokaryotes. And at this point, we understand what prokaryotes are. This is in reference to the cells. Remember the lecture about cells? We looked at those that are found in the eukaryotes, such as us, and those that are found in bacteria, and these are referred to as prokaryotes. So right from the start of this lecture, I want us to understand our focus when we look at DNA replication. So what are some of the learning outcomes? I've included this slide. It wasn't part of the notes that I sent, but I'm hoping that in your own time, because this is a makeup, you're going to go through all these points so that by the time you're finishing this topic, you would have understood what is expected of you. And I don't want to go through them, but I'm hoping that in your own time, please go through this slide, do a checklist, and make sure that you've understood all these. Okay, so I want to start by looking at the understanding of what DNA replication is. Remember, prokaryotic cells are single-celled organisms like bacteria. And these cells multiply to make more of themselves. When you think of DNA replication, and also in relation to these bacteria cells multiplying themselves, that is making more cells of themselves, they need to duplicate their DNA. So when you think of how you can define DNA replication in prokaryotes, it is a process by which the genetic information that is found in the prokaryotic cell is duplicated before the cell divides. So what you see here is what is referred to as a mother cell, and you can see it is just one cell. And in there, we can see some kind of ribbons. At this point, they're representing genetic material. But then we also know at this point that in prokaryotes, this DNA is circular, it's not linear. So while this diagram is showing these chromosomes as though they are linear, we should keep in mind in prokaryotes it must be a circle. And what this means is that before a cell can divide, it will first of all duplicate its DNA. And by duplicating, I mean making an extra copy of itself that is exactly the same. It's like creating a clone of myself. If you want to clone me, everything about me must be exactly the same. So DNA replication, the end product, in this case, after duplication, should be the exact copy. This understanding is very important because some of the questions you'll be given a strand of DNA and you'll be asked to find which one of these is a daughter strand. The daughter strand must be the exact copy of the parent strand. In this case, we have a mother cell. When this cell duplicates DNA, the duplicates must be the exact copy. And by exact copy, guys, I mean the combination of nucleotides. The A's, the T's, the G's, and C on this strand must be the same with what was on the parent strand. And if this is one strand, whatever the combination was, the way the A's, T's, G's, and C were arranged, it must be the same exact information on the daughter strand. And also, before I move on, I want to make this very clear. We must understand, where is DNA replication taking place? And this time around, we're referring to a prokaryotic cell. We know that their DNA, or should I say, the cell does not have a nucleus, do not have membrane-bound structures. In short, they don't have a nucleus. So if we're talking about DNA replication, we must think of, where is this material found? From our cells, we know this information is found in the nucleoid region. For that reason, DNA replication is taking place in the nucleoid region. The nucleoid region is found in the cytoplasm. It is not a membrane-bound structure. So the take-home message, the end product of DNA replication must be the exact copy of the parent molecule. In prokaryotes, DNA replication is taking place in the nucleoid region because this is where the genetic information is found. They don't have membrane-bound structures. So moving on, we want to now look at the process of DNA replication. The process itself is divided into 3 major steps. 3 major steps, not to say there are not other steps, but these are the major ones. We can collect them into 3 major steps. The first step is initiation, and of course it has got all this. We're going to look at each one of them. The next one is elongation, and then termination. In short, initiation is the starting of DNA replication. Elongation, you're adding nucleotides and these are being, the nucleotides you're adding, the information that is going to guide the addition is going to be from the mother strand, the parent molecule. And then finally, you have to terminate. So think of it like a process of initiation, elongation, and then termination. Now let's start with initiation. Now, up to this point, we know that DNA is a double-stranded molecule. And this molecule is carrying a lot of information. Remember, I even mentioned in the previous lecture, in the introduction to this course, I spoke about a gene as just a segment of DNA that is carrying information for a particular function. Now, in replication, they are going to make a copy of the genetic material, everything that is found in a cell, not just a section. So DNA replication is replicating the entire genetic information in a cell. Keep in mind, the DNA is double-stranded. Now, initiation, or should I say, DNA replication has to start from somewhere. That space or the segment on the DNA where replication initiates, where replication starts, is referred to as the origin of replication. We're saying that DNA replication, or should I say initiation, has to start from somewhere within this DNA molecule. And like I said, it is circular. So the way it has been depicted here, it's like it's a linear molecule. But keep in mind, bacteria genetic material is circular. Now, where around that circular molecule does DNA start? The region or the segment on the DNA molecule where DNA replication starts is referred to as the origin of replication. So what you see here is an origin of replication, and then from there, this origin of replication is unwound. It's like opening up a zip, but not the whole section, just a single region is opened up. And in that region, you're going to find that the DNA molecule is no longer double-stranded. We have single strands in that region. Why is this very important? The first thing you should note here is that DNA replication starts from somewhere. That region is referred to as the origin of replication. And of course, if you're looking at bacteria, we refer to it as the ori-C. The ori-C is the origin of replication, and the C there just stands for chromosome. That's in bacteria. This region is important because this is where replication initiates. But what is happening there, that is very important. That origin of replication allows for the cell to create strands that are single-stranded. Why? Because DNA replication can only replicate or can only take place on single strands. But that is not the nature of DNA, isn't it? It is double-stranded. So first we have to create a region where there must be single strands. And this region is the origin of replication. In bacteria, it is referred to as the ori-C. Now, what are some of the features of this region that we're referring to as the origin of replication? So, what I'm asking you here is, if DNA is double-stranded, and we know that those strands are held together by complementary base pairs, where when you have an adenine, an A, it must base pair with a T with 2 hydrogen bonds. If you have a guanine, it must base pair with a cytosine with 3 hydrogen bonds. Now, for the cell to break those hydrogen bonds, it uses energy, and it's going to be easy for it to break a region that is rich in A's and T's compared to one which is rich in G's and C's. Why? Because a region that is rich in A's and T only has 2 hydrogen bonds between those nucleotides, and it will be easy to break compared to a stretch that is rich in G's and C's. What I mean by that, you can think of this region as having a rich AT combination, meaning that you've got more than one adenine and thymine in that region. That's why what I mean when I say rich. It has got more nucleotides that are in the form of A and T compared to G's and C's. And it is good for this region because it allows this region to be easily unwound for the hydrogen bonds to break in short. Another thing that the origin of replication has is what is referred to as a DNA recognition site, also referred to as a DNA box. Now, the origin of replication, just like any other region in the chromosome, has got nucleotides, A's, T's, and everything else. So the cell has to find a way to identify this site. And this site has a marker. And one of the markers that you see there is a DNA box. What I'm trying to say here is you should think of it. The chromosome itself, all the way, is a combination of different nucleotides. But then the cell has to identify which one of these nucleotides is a point where we start. Or in short, which one of these nucleotides has got the origin of replication. One of the features is, of course, it is rich in AT. Another feature is that it has got this recognition site that is referred to as a DNA box. This site is a site that another protein comes in to identify the origin of replication. Or in short, the protein now that comes in to identify this site is referred to as DNA. This is a protein that is referred to as an initiator protein. Or in short, what I'm trying to say here is that the cell has a protein that is known as DNA. It is an initiator protein, and its function is to recognize the DNA box in the origin of replication, kind of marking it to say this is where DNA replication should start. Now, let's look at how this origin of replication is oriented. I want us to focus on this diagram here. Like I said, it is represented here as a linear molecule, but keep in mind it is secular, meaning down here there's more DNA sequences. At this point, we have just extracted the origin of replication, and we want to find out what features does it have that allow it to serve as an origin of replication. One of them I've already spoken about. which is the one that has the DNA box. So the origin of replication has got two sites. One of them is the site where the DNA protein binds, and this one is referred to as the 9-mer. The number 9 just represents the number of nucleotides that are in that section. And there are 4 of them. So you've got one 9-mer here, another 9-mer here, another one there, and another one here. This region is just referred to as the 9-mer, and it is a region that initiates the identification of the origin of replication. And when I mean identification, it is the region that the protein DNA is going to recognize as the origin of replication. In addition to that, the origin of replication has another sequence that is referred to as the 13-mer. The 13 in there just refers to the number of nucleotides that are in each of those yellow boxes. So then, the origin of replication has got two sites within it. The first site, the function is for recognition, and the protein that helps to recognize this region is referred to as the DNA A. The origin of replication also has another site within itself, and this one is referred to as the 13-mer. The number 13 is just a number of nucleotides that are in each one of those boxes. The 13-mer has got 3 segments. So one 13-mer here, the second one, and this is the third. So collectively, the origin of replication has a 9-mer, and there are 4 of them. Then it also has a 13-mer. What is the function of the 9-mer? The 9-mer, its role is to act as the identification point of the origin of replication, and it binds DNA to ensure that it has been recognized. What is the function of the 13-mer? The 13-mer is a part of the origin of replication that is unwound, like opened up, and it is rich in a combination of A's and T's. And the reason is that it is easy for the cell, for the machinery of DNA replication to open up a region that is rich in A's and T's compared to G's and C's. I hope this is clear. And one more thing that I want to mention, I've included this slide deliberately, is that in both prokaryotes and eukaryotes, shown in this picture, the prokaryotes, we know their genome or their genetic material is circular. Ours is linear. The genome always has an origin of replication. In bacteria, the prokaryotes we're looking at, they only have one region of origin of replication. But for eukaryotes such as yourself and me, we have got multiple origins of replication. So what this means, for example, when I say multiple, consider this as a stretch of DNA in our cells. Of course, this will be like one chromosome. That one chromosome is going to have multiple sites where replication can take place. So let's say, for example, I hurt myself, and that portion of my wound needs to be sealed by other cells. Before those cells can be formed, the DNA around the healthy cells has to be duplicated. And this replication is going to start at multiple sites. In short, eukaryotes like you and me have got multiple sites of DNA replication, and this is, it serves a very good purpose because our genomes, we've got a lot of genetic material. Our genomes are large compared to these guys who are very simple. Okay, so this is very important for us to understand. In prokaryotes, you're dealing with only one origin of replication. In eukaryotes, there are multiple regions. So now, I'm going to go back to the prokaryotes. I thought of just mentioning this because it's very important for us to understand that we're dealing with only one region of replication, and replication will take place when it opens up in both directions. We'll get to that. So at this point, I want us to start with what happens when the DNA recognizes the 9-mer. Remember, I said the origin of replication has got a 9-mer on one end and a 13-mer. The first point is to recognize the 9-mer. So when DNA protein, which is referred to as the initiator protein, binds to the 9-mer, it causes a conformational strain. So in this case, imagine DNA is a double-stranded molecule binding to themselves, and then suddenly you add another protein, which has got a very good affinity to bind to DNA. Because of this binding of DNA to the 9-mer, it causes a conformational change, like a strain, that allows for the region in the 13-mer, which is rich in ATs, to unwind, to open up, or in this case, for the hydrogen bonds to break. Now, at this point, we have single strands along the 13-mer. What has caused that? The binding of DNA to the 9-mer. Now, at this point, I want us to understand something. The DNA is not an enzyme, no. It's a protein in nature, and it has got a high affinity to bind to the 9-mer, because remember, the 9-mer has got a DNA box, like a recognition site where these guys can bind. So the unwinding of DNA in initiation is not facilitated by active unwinding, like an enzyme. No, it is facilitated by a conformational strain that is caused by DNA binding. Now, to keep unwinding DNA actively, we need an enzyme. And that enzyme is DNA B, or should I just say helicase. Helicase is the enzyme that actively unwinds DNA. What I mean by that is that for the helicase to continue to come and bind to DNA and unwind the DNA, it needs a single strand. So because DNA initially was a double strand, helicase cannot function. But it can be activated to function once there is a loop, or should I say in this case, single strands, which have been made possible by the binding of DNA. So one thing to note here is that DNA causes a conformational strain, which leads to unwinding. Because in this region now we have got single strands, helicase can join or bind to this DNA and now actively unwind the DNA. I'm using the word actively because the enzyme helicase uses ATP. It uses energy to break the hydrogen bonds. And now it is, if you think of how the body is made, and every time when I say you guys must eat shellfish to have more energy, these processes require energy. In short, DNA replication is an energy consuming or demanding process. Okay, so now at this point, the understanding is that we now have single strands of DNA, and these single strands of DNA can function as a template. And these single strands are there because we have the enzyme helicase. I want also to mention that, yes, despite us having these single strands, because helicase has opened them up, they can still complementary base pair. Because remember, that's how they were in the first place before you unwound them. To keep these strands single-stranded, proteins are recruited to the DNA strands. These proteins are referred to as single-stranded binding proteins, in short, SSPs. So the function of single-stranded binding proteins, one, is to bind to the single strands of DNA to ensure that they don't go back and complementary base pair. And this is very important because DNA replication can only function when you have single strands. I don't know whether that's a question. I can't see. Someone has their mic not muted. But anyway, let me continue. If it's a question, you might want to stop me, but we don't have so much time. So I'll move on to just mention a few things about the enzyme helicase. A helicase, sometimes it is also referred to as DNA-B in bacteria, is the enzyme that actively unwinds DNA because it's using ATP, so it is actively unwinding. Now, the helicase binds to only the strand that is in the 5 to 3' direction. Remember, DNA is antiparallel. One strand runs from the 5, and then the other strand from 3 to 5. This is antiparallel. So which one of the two strands is a DNA helicase loaded on? It is loaded on the strand that is running in the 5 to 3 and not the strand that is running from the 3 to 5. For the simple reason that the helicase has polarity. It can only move in the direction 5 to 3. And I hope you know what that means. When I say 5, it means I'm standing at this position and I'm moving in this direction. This other strand is moving from the 3 to 5. We know if this end is 3 prime, this end here should be 5 prime. So the helicase of these two strands, it loads on the 5 prime strand, the one that is moving in this direction. Now, from here, I hope you've understood which of the two strands the helicase loads on. It's not a decoration that it is loaded on the 5 and maybe you're going to find it on the 3. Yes, some diagrams will show that, but no, helicase has a polarity. It can only choose one of the two strands and not both. In short, it's not binding to both strands. It's only binding to one of the two. So what do you have here? You have your helicase going this way. And of course, behind here, you should have those single-stranded binding proteins to keep these unwound strands, independent. So I think this I've already mentioned. This is just trying to emphasize what is happening at the origin of replication. You have your DNA A binding to your 9-mer, and this causes local unwinding in the 13-mer. Because now there's an opportunity for single strands, helicase comes in and binds to one of the strands, and this strand is going to be the one running from the 5 to 3. So in this case, it should be running in this direction. This is where this one should be going. And in this one, you've also got another helicase binding to the two strands. Now, keep in mind, helicase is only binding to one of the two strands, meaning of the two templates, there's helicase on this strand running from the 5 to 3, and there's also helicase on this strand running from the 5 to 3. What does this mean? It means DNA is going to be opened in both directions. And that is why at some point it makes sense to say DNA replication is bi-directional, because right from the start you have DNA being unwound in both directions. Okay, so now what is a take-home message from here? DNA helicase unwinds DNA in both directions, meaning that if the origin of replication was at this center, you have opening of DNA in this direction and opening of DNA or unwinding in this direction. What this means is that we are going to have two replication forks. That is the region that has been unwound and will allow for active DNA replication. On this other end, you also have another region where DNA is actively being unwound and is going to allow for DNA replication to take place. So in short, at this position, you have the origin of replication, and because helicase is unwinding DNA in both directions, we're going to have two replication forks. So what do we have now at the origin of replication? Guys, if you have your mic on, you're creating an echo on my end, kindly mute your mic. So now, I want us to focus on this diagram. Remember, this dot here is the origin of replication. And remember, it has got a 9-mer and a 13-mer. After unwinding in the 13-mer, we said DNA helicase, or should I say helicase, is recruited on the strand that is running in the 5 to 3. And at this point, we understand replication will be in both directions. Now, this region is what is referred to as a replication bubble. The replication bubble is going to have two replication forks. Remember, DNA helicase going in this direction and in that direction, creating a Y-shaped, which is referred to as a replication fork. So a replication bubble has got one origin of replication and two replication forks. The replication fork is a region consisting of single-stranded DNA or DNA which has been unwound and is going to allow for active DNA replication. The replication fork is just a region within this replication bubble where the two strands of DNA have been opened up or unwound, and because they've been opened up, they can allow for DNA replication to take place. Now, if you've understood this, I now want to move on to the single-stranded binding proteins, because we are saying that for DNA replication to take place, both strands, we need a template, and the template must be single-stranded. So now, the single-stranded binding proteins are the ones that are ensuring that as the helicase unwinds, the strands behind them do not anneal, do not complementary base pair with each other. So what you see here are single-stranded binding proteins attached to both strands, because remember, they can complementary base pair. So one of the main functions of the SSBs is to stabilize the strands so that they can remain as single-stranded. Another reason is to ensure that this open strand is not degraded by enzymes. Remember, one of the reasons why RNA is very active is as you see, it's a single strand. So the moment you open DNA, you are exposing it to everything that is in the cell, including nucleases. These are enzymes that break down DNA. So one of the functions of the SSBs, yes, is to ensure that the two strands are stable, remain separate as single strands. The other reason is to protect them from degradation so that they don't get, damaged and things like that. The next slide is just trying to emphasize everything else that I've spoken about. And I now want to move on to topoisomerases. On this slide, what are we looking at? Remember, if the helicase is unwinding DNA in both directions, there's going to be strain upfront. Think of yourself holding a loop that has been, for example, a thread that has got two ropes and they have been intertwined, and then you decide to pull one section. When you start pulling at the beginning, it won't be very difficult, but as you continue unwinding that loop, there's going to be strain on either side. And in DNA replication, this also happens. As a helicase is unwinding, up front there's going to be strain. Now, if this strain is not dealt with, it is not relaxed, it's going to push back, and this machinery here is going to fall off. Now, to prevent, or should I say, to relax the DNA, preventing the supercoiling and the strain that is coming because of this unwinding, another enzyme or other enzymes are recruited. These enzymes are referred to as topoisomerases. So topoisomerases, also referred to as DNA gyrase or gyrase, these are enzymes that come in to relax DNA. And by relaxing, I mean they prevent supercoiling of DNA or the strain that is caused by helicase unwinding of DNA. In this case, just imagine yourself holding a rope where they're intertwined in two. You open only a small section. When you start, it will be easy, but as you're gone pulling them apart, and in DNA replication, we don't want that. Otherwise, everything else back here is going to collapse because of the pressure, the force coming from the unwound regions. So this is just a summary of everything that you have at the replication. And we know what they're doing there. It's actively unwinding. And in this diagram, you can see it is shown as if it is bound to the helix to 2, but at least we know it is bound to the 5' of the strands and the topo. These ones, they help to relax the DNA so that as helicase is unwinding, it's not finding it so difficult because of the pressure of unwinding both strands. I want us to understand that everything else we have covered, this is just initiating DNA replication. You haven't even started replicating it. It's trying, in fact, one of the most important things in initiation is to get single-stranded DNA, because you cannot elongate, you can't add anything to it if you don't have those two strands of DNA. Now, at this point, you have your single-stranded DNA, and now you want to elongate. DNA elongation is just a process where you're adding nucleotides to DNA to make a new molecule. And this one requires you to go back to your notes and look at the nucleotide, its components, or should I just say its features. Okay. In fact, it will be easy for you to understand and not even memorize. I always encourage you, do not memorize, just understand. if at this point you don't know the structure of a nucleotide, you might want to go back there, because in elongation, we are adding those nucleotides to make a new strand. One thing we should mention here is that DNA replication or DNA synthesis, or in this case, elongation of DNA, should take place in the 5 to 3 prime direction. And this orientation here is coming from the nucleotide. Okay, because we know on one end of the nucleotide, we've got a 3 prime end on the pentose sugar, and on the other end at the 5 prime, we have a phosphate. So this orientation is coming from the structure orientation of a nucleotide. Of course, we know it is a direction of synthesis, but it is also guided by just the structure of a nucleotide. So now let's look at DNA elongation, in this case, the process of elongation. So what you see in this diagram is a template. And by the template here, first of all, the understanding is that it is that region of DNA at the replication fork that is now a single strand, has got those SSBs stabilizing it. And we know what a template is. A template is just a pattern that you can use to make an identical molecule. So this template means that whatever we're going to make out of this should be the same exact copy. And that is why DNA replication leads to the synthesis or formation of the same exact DNA molecule in the daughter cells. So now here you have your template and you understand how this came about. Okay, and keep in mind, this process is taking place at the replication fork. And because we've got 2 replication forks, it means it's taking place on both of them. So at this point, I'm not showing both replication forks, I'm only showing one. But keep in mind, it's taking place at the other one as well. So in DNA replication, we are adding nucleotides. So what you see here is a stretch of nucleotides. At least we can see 3 of those have been added. 1, 2, 3. These are 3 nucleotides. How are we counting them? Because we know this one here is 1 nucleotide. This is 1 nucleotide. This is another nucleotide. And at this point, we want to add another nucleotide. The nucleotide that is coming in, you can see it has got guanine. How has that been guided? Because on the template, we have a C. And because DNA synthesis obeys complementary base pairing, what should come and bind must just be a G. It cannot be an A, it cannot be a T. No, they're not complementary base pair. And how is it being added? If you look at this nucleotide, on this end, this is the 3 prime end. And on this end, this is a 5 prime end. Like I said, if you look at your nucleotide and you've understood it, you know why this is a 3 prime end on the pentose sugar. It is the numbering of the pentose sugar. And this one is a 5 prime end. The nucleotide that is going to be added to synthesize the new molecule is added from the 5 prime, or should I say using its 5 prime, and it will be connected to the 3 prime of the already existing nucleotide. DNA synthesis is from the 5 to 3. If you look at your nucleotide, this is a 5 prime end. The 5 prime end of your nucleotide will be attached or bind to the 3 prime end of the already existing nucleotide. Now, I also want to mention this process is catalyzed by DNA polymerase 3. The enzyme that is going to allow the binding of this nucleotide to the already existing molecule, the new template, is DNA polymerase 3. Now, one of the limitations of DNA polymerase 3 is that, yes, it can allow for synthesis of DNA, the addition of new molecules in the 5 to 3 prime direction. But this enzyme is not very intelligent. It cannot initiate synthesis on its own. It needs a short sequence to be existing already before it can do it. What I'm trying to say here is that DNA polymerase 3 is the enzyme that catalyzes the synthesis of the new daughter strand in the 5 to 3 prime direction. But there's a limitation here. It cannot initiate synthesis on its own. It needs a pre-existing strand. Now, this short strand that is synthesized to enable DNA polymerase 3 to continue synthesis is referred to as an RNA primer. In fact, it is somewhere here in your note. A short RNA primer is synthesized through complementary base pairing. And this short RNA primer, what does it do for DNA polymerase 3? it provides a free 3' OH group that can allow for the 5' of the new nucleotide to be added to the chain. An RNA primer has to be synthesized first to allow DNA polymerase synthesis of new strands. This RNA primer is RNA in nature, meaning that these nucleotides are RNA. So maybe here we should have had the uracil because there's no U in RNA. What this means is that DNA replication, or should I say elongation, is initiated by an RNA primer. Once the RNA primer is positioned or has been synthesized through complementary base pairing by, of course, an RNA polymerase, we'll look at it in the next slide, that is the only time when DNA polymerase 3 can come in to elongate DNA. So meaning that DNA polymerase 3, yes, it is an enzyme that catalyzes DNA, polymerization, but it is not too intelligent to start on its own. It needs a primer. That primer is RNA in nature, meaning that the nucleotides in the primer will be RNA in nature. They'll not be deoxyribonucleotides. They'll be ribonucleotides. And what that means is that they will have an OH group on carbon number 2. So when you're given a strand and you're asked to fish out, to count the number of primers, it will be very easy for me to tell how many primers are there or points of primers, because I'll know the region that has got uracil should be a region where the primer is. Okay, so moving on from here, let's look at the primer that is synthesized. What you're seeing here is just one end of the replication fork. Remember, there's another one on this other end. Because of space, let's just use one of them. In fact, let me use this slide. So think of it that we have blown this slide to show only one end of the replication fork, and the origin of replication must be somewhere here. We are saying that for DNA polymerase to start elongating DNA, it needs a primer. That primer is synthesized by an RNA polymerase that is referred to as a primase. So primase is an enzyme that synthesizes the primer. Its nature, it's an RNA polymerase. It's not a DNA. Remember, we've got DNA polymerase. This is RNA polymerase, meaning that it is RNA in nature, or should I say it synthesizes ribonucleotides. In short, the primer is synthesized by the enzyme called primase. Primase is a form of RNA polymerase. Now, let's look at the two strands. This strand of DNA is the 3' end. This strand here is a 5' end. And we can clearly see that on each of these two strands, we've got primers. The primase has synthesized this short stretch here, and it has also synthesized this short stretch here. Let's look at the direction of this primer. Let's start with strand A, which has got the 3' end. So, synthesis of the primer also obeys the 5 to 3' direction. Synthesis of this primer by RNA primase also obeys complementary base pairing as well as the direction of synthesis. It must be from the 5' end to the 3' end. So when you look at this strand here, this is the parent strand. This is the primer. The primer, you can see, is synthesized in the 5 to 3' direction, meaning you are standing at this position as the 5' going this way. Why? If this strand is 3', the complementary strand must be antiparallel. This must be 5'. Let's go to strand B. Strand B at this end is 5', and the complementary strand should be 3'. But we are saying that the direction of synthesis of the primer by the RNA primase must also be from the 5 to 3, meaning that this primer will be synthesized in the opposite direction on strand B. Now, let me use the replication fork as a reference point. So I'm going to say that one of the primers is synthesized in the same direction as the replication fork. And by that, I mean the replication fork is going in this way. This is how DNA helicase is opening up, isn't it? So the fork is going in this direction. One of the primers is synthesized in the same direction as the replication fork. And this has got a consequence of what we're going to call the product of what comes afterwards. that is to say the leading strand. The reference point will be the replication fork. And by that, I mean the helicase is opening this way, so the fork is going in this direction. Strand B, this primase is synthesized in the direction away, in the opposite direction to the replication fork, or should I just say away, meaning it was synthesized from this end going this end. So now, let me move on to the key features of elongation. I've already mentioned them though. We know we have the enzyme DNA polymerase 3 as the one that synthesizes DNA, and this enzyme is not very intelligent. It always requires that for it to add another nucleotide, it needs a free 3' OH group. And this one we know is provided by the enzyme primase, which is a primer. DNA polymerase 3, we're also going to see that one of the strands is referred to as a leading strand, and this is the strand that is synthesized in the same direction as the replication fork. And the one that is synthesized in the opposite direction, replication fork, we're going to refer to as the lagging strand. Neither of these strands are going to exist without the primers because we know the enzyme is not that intelligent. In addition to that, we're also going to see that DNA replication, or in this case elongation, is not immune to mistakes. It also makes mistakes. And when it makes mistakes, it can correct itself. To correct itself, it uses this activity that is referred to as 3' to 5' exonuclease activity. Now, let's look at just what 3' and 5' means. It means you must stand at the 3' end and move in the direction of the 5'. That is why it's referred to as 3 to 5' exonuclease activity. Exonuclease just means that you are digesting or degrading, the nucleotides, destroying in short. So 3 to 5', we are destroying these nucleotides that are wrongly placed by moving from the 3' to the 5' direction. So at this point, I want us to look at the replication fork, first of all, as one whole structure. And then from there, I'm going to cut it into two, just for illustration's sake. So what you see here is an origin of replication. Remember, the 9' and the 13' are all sitting somewhere here. Once you have your single strands as templates, the helicase comes in to actively unwind your DNA, creating two replication forks. So meaning that this replication of origin now becomes a replication bubble with two replication forks going in opposite direction. And because DNA replication is going to take place on this replication fork and on this other replication fork, we say that DNA replication is bidirectional, meaning it's going to take place in both directions. So now, allow me to break this replication of origin so that we can understand how replication is taking place, by not looking at both at the same time. So I'll start with only this replication fork, then I'll go to this other replication fork. So this replication fork is just one of the two. I've just split that diagram into two. The first thing that I want us to note here is the orientation of the parent strands. The parent strands are in blue. Strand A, which I've just designated as A, is from the 3' going to the 5'. Strand B, at this end, because DNA is antiparallel, this should automatically become the 5' and it is going to the 3'. Let's start with strand A. How do we synthesize this strand? Remember, the first thing we should have is a primer. Sorry guys, I needed to collect something. Let me just pick up a call. I apologize. Guys, I apologize for that. I can see I just have, not so long. But anyway, if I explain for this end, I'm sure the other end, you must also be able to understand. So if we look at strand A, running from the 3 to 5' direction, want to find out how is this strand going to be synthesized. Is it going to be synthesized in the same direction as a replication fork or in the opposite direction as a replication fork? That's what we're trying to figure out here. So how do we do that? So synthesis should be from the 5 to 3 prime. If this end is the 3 prime on the parent DNA, then we can synthesize a primer and allow for synthesis to take place from the 5 going to the 3 prime. Therefore, the strand that is synthesized in the same direction as a replication fork is referred to as the leading strand. And this strand will only need 1 primer to start. By that, only 1 primer is needed, and after the primer is made, the DNA polymerase 3 can continue synthesizing this strand. And by synthesizing, I mean making a new molecule using the information that is on the template. Let's go to strand B. Now, strand B, at this point, this is a 5 prime. Now, synthesis cannot run from here because this will mean if synthesis should run from here, this strand would have a 5 prime, right? But then, because synthesis is from the 5 to 3, it will mean that the primer will be made upfront so that this end will be 5 prime to 3 prime. And when it reaches this end, we're going to have a complement, an antiparallel strand, the 5 prime here and the 3 prime. In short, one of the strands is going to be synthesized in the opposite direction to the replication fork. What I'm trying to say here is that strand B is from the 5 prime to the 3 prime. Because synthesis must take place from the 5 to 3, a primer will be made internally on this strand, and that primer can now allow for synthesis to take place from the 5 prime to the 3 prime. So now, the strand of DNA which is synthesized in the opposite direction to the fork is referred to as the lagging strand. Now, what this means is that on the lagging strand, you're going to have more than 1 primer because synthesis is taking place internally. For example, in this case, it didn't start from here, it started from internal on the strand. Once this stretch is synthesized, it will fall out, stop. Again, another primer will be made to allow for synthesis of a stretch. Now, keep in mind, these are not as small as they look. This could be about between 1,000 to 2,000 base pairs. Of course, for illustration, they look short. But for each new synthesis on this strand, it will be interrupted. And the short strands that are synthesized are referred to as these Okazaki fragments. What this means is that each Okazaki fragment is going to have its own independent primer, and so on and so forth. Or, in another way, we can say that on the lagging strands, we have multiple primers. While on the leading strand, you only have 1 primer because it is synthesized continuously. This one is synthesized discontinuously in the direction opposite to the replication fork. So now, remember I mentioned that this is just one end of the replication fork, meaning that what I've explained on that strand, on that end, must be the same on this end. So if replication is starting from here, this is the replication fork, it means that this strand here should be synthesized in the opposite direction. The primer will be made internally to move from the 5 to 3. And this one will be our lagging strand. This one here, because this strand is a 3 prime end, we can just have 1 primer and synthesis can proceed in the same direction as a replication fork. And this becomes our leading strand. What is important for us, guys, let me use this strand, is when you're given an origin of replication like this one, or should I say replication bubble, you must be able to designate, to show which one of these is a leading strand and which one of these is the lagging strand. So at one replication bubble, you're going to have 2 leading strands and 2 lagging strands. But how you come up with this, the best for me is to split this replication fork and then designate the orientation of the strands. And I hope this has been clear or cleared out. So this is just a note for you. This is something that I'm hoping you can go and look at in your own time. Given such a strand, nothing is given to you. Can you tell which one of them is a leading strand, which one of them is a lagging strand? Of course you know. If a strand has got multiple primers, you know this is being synthesized away or opposite the replication fork, and this becomes a lagging strand. If it is being synthesized in the same direction as a fork, then I know this is my leading strand. But take your time when you don't have so much to do. Just look at it so that you can familiarize yourself with this concept. Is that a hand?
Yes, madam. There's a group that's supposed to attend the class at 9.
Okay, I can end here.
Yes.
Yeah, I don't know when I can finish up, but I think, like Mike advised, this lecture is on it was streamed, isn't it? But if you still need me to help you understand, I think you can let me know. We can always schedule something. Thank you very much for making time for this. We'll see you Monday. Thank you, Mike, for facilitating this meeting. Guys, have a good day. Bye.